here in Newport. He also is courtesy faculty, at least at the Department of Fisheries and Wildlife. So he works with a lot of our students as well. His research focuses on the processes that control variability of marine ecosystems and the transfer of energy from planktonics to fishery systems. And so we're going to learn a little bit more about some of that. A little bit of background on where he went to school. He did his bachelor's in fisheries at the University of Massachusetts, his master's in oceanography at Oregon State University, and his PhD at the University of Washington. And as my voice fades away, I'm going to hand it off to Rick. Thank you so much for coming. I'm going to turn one of the lights down a little bit lower, but not both.
Thanks, Cinnamon. And thank you all for coming. I'm really impressed that there's this many people that are interested in pyrosomes around here. I'm totally shocked, actually. Anyway, what I'd like to do is talk a little bit about some recent research we're doing. Well, one of the things before I get started is, although my name is the only one that showed up here on the front page, there's actually, this is part of a very big group effort, and I wanted to acknowledge a number of people that have contributed, and you'll see their names in certain parts of the talk, but there's really a whole bunch of people that I've been working with over the last few years on pyrosomes, and there's just a listing in lots of different departments, mostly at Oregon State, but other University of Oregon and ODF&W as well. So I wanted to thank all these people for their assistance. So a lot of you were probably wondering, what is a pyrosome? And I think if you were going to ask me that 5 years ago, I probably would have said, I have no idea. Or, I mean, I've heard about them, but I had no idea what they were because I'd never seen one before, even though I've worked with a number of, worked in the system for probably 30 years now and worked in a lot of different kinds of jellyfish, but I'd never seen a pyrosome in my life. But this is an example of probably how most of you may know what pyrosomes look like, is that they washed up on the beach in huge numbers the last few years, and they kind of look like these transparent or translucent pickles, you know, and they're just everywhere on the beaches. So that's one of the things that most people kind of are aware of in terms of pyrosomes. But in reality, they're actually a very important group. They're one of several gelatinous zooplankton, and you're probably familiar more with the ones up here in the upper left-hand corner, which are your typical jellyfish like the sea nettles and moon jellies and things like that. You might be familiar with the comb jellies, which are the ones in the ctenophores in the lower right-hand corner. But the other four, those two groups are actually very primitive, some of the most primitive invertebrates out there evolutionarily in the world. But these other four are part of a group that are called the thaliaceans, and they're actually very advanced. They're one level below the vertebrates. They're actually chordates, so they have a notochord, but they're not actually all the way up to, they don't have a regular backbone like the vertebrates, but they're the next step below it. So they're actually quite different in terms of their evolutionary history. So these are what we'll be talking about today, pyrosomes. And just to kind of put them in perspective, you have sort of a bunch of different groups in these tunicates, and the ones that probably most of you are familiar with are actually on the bottom. They're called sea squirts or ascidians, and those are very common in a lot of ecosystems. And so they're closely related to these other groups here. The second one are called the larvaceans or the apodicularians, and this turns out to be the most closely related invertebrate to all the vertebrates genetically. And then there's also this other group that are called the thaliaceans, and there's a bunch of them that are things called salps. You've probably seen some of those washing up on the beaches or heard about them, doliolids. But what we're talking about today are pyrosomes, and there's actually, it's probably one of the least species of these groups. There's only about 23 species that are found throughout the world. But there's one that's over and above the most important, and that's the one I'll be talking about today, this Pyrosoma atlanticum. This is one of the dominant pyrosomes in the world's oceans, and it's mainly found in tropical and subtropical waters, you know, so out in the middle of the ocean, very blue water, as you can see from this picture. And it actually has been known for a long time. A lot of the early explorers, mariners that used to see these large bioluminescent blooms all throughout the oceans, and they first started, you know, going to the tropics. And it turns out that the pyrosomes are one of the main causes of that. And the name, in fact, pyrosoma is Greek for fire body. So that's where it comes from. So they're actually very highly bioluminescent. So this is a map of the distribution of this species, and it was, according to this author back in 1981, they were found mainly between 50 degrees north and 50 degrees south. But as you can see in this figure, most of those ones up further north or up in the North Atlantic, it is warmer for a particular latitude compared to the Pacific. In the Pacific, they've only been really found as far north as Northern California or Southern California, maybe straying up to Central California, but never up in our area, which is shown here as that star. So the reason that is because our water is much too cold and much too productive for these guys. They really don't like the typical water we have here in the North Pacific. However, things have really changed in the last few years, and that is due to a very unusual event that happened, a lot of you are familiar with. What we have here is what's the sea surface temperature anomalies. So these are the differences from the long-term averages quarterly from June 2014 to August 2017 down the lower right-hand corner. And you can see the temperature scale on the bottom. So the reds are above average. And you can see here that throughout this whole period there's a lot of red there. The ocean has been very warm in the Pacific. And that's been caused by basically two different things. There was a initiation of what was called the warm blob that a lot of you have heard about, which is a really unusual event, but it started sometime during the winter of 2013, 2014, out in the middle of the North Pacific, and then started moving out and growing and moving out throughout the whole Pacific. And you can see it as it's moving closer to shore. It actually came on shore in the fall of October 2014. And then for the next year, it pretty much dominated the system. And then what happened was there was a tropical El Nino that started down off the coast of Peru and Ecuador. And then that moved up the coast and it ended up by having very warm temperatures for about a 2 or 3 year period here, much warmer than we normally get. And to look and see how things changed so dramatically off here, this is from sea surface temperature anomalies from off a buoy that's about 20 miles off the coast here. And you can see sort of you get a seasonal pattern where you get upwelling events that drop the temperature, the blue part there, and then other times it warms up above average. But look what happened around September 14th of 2014, I think around 10:30 to be exact, this warm blob came over the buoy and ended up by elevating the temperature about 6 degrees centigrade, which is really incredible. I mean, it's more than sort of the seasonal change that you get overall. And it literally just completely changed the system. And you can see it slowly started getting cooler, but not really. It's still been well above the average for the rest of that year. So this is really a big change. And we're getting peak temperatures of 19.4 C, which is really, really warm for this area. And this is a plot showing sort of the multi-year trends. And what we see here in the black line is the sea surface temperature within that box there. It's outlined off of the coast of Washington and Oregon. And what you see is the sea surface temperature is the black, and then the blue line is sort of the long-term mean. So how, the sort of long-term average. And the area that's colored in yellow are areas that are times that it exceeded a certain threshold, one standard deviation above average, which is what the threshold is for what they call a marine heat wave. This has been established in a lot of different areas, and they do have marine heat waves all around the world. But this one has turned out to be one of the longest ever recorded. It lasted for about 711 days off a year, and it's also one of the most severe, not the most severe. They've had some stronger ones off of places like Australia, but it was considered to be severe, which is sort of like a Category 4 hurricane at that level. It's just right below the most severe. So this is a really big event that happened and really changed the ecosystem off here. So it led to what we call the pyrosome bloom, and this is what I'll be talking about today, giving examples about what happened and what we saw during this bloom. So one of the things I did want to mention, though, is that it has been picked up a lot by the media, and in fact, once when these things started washing up on the beaches and the fishermen were getting word out there that they were catching all these guys, they really ended up by being it kind of spiraled out of control, where I was like coming into my office and I was getting phone calls like 3 or 4 a day about what's going on out there, and it was just amazing how much it really got picked up. So it actually got to be, I was getting calls from Europe and New Zealand and things like that. So it made the real international news. So it's kind of interesting. So I'll give some examples of how we detected this and where we started seeing this. And this is from some surveys that I'm involved with, where we go out and sample within that colored area there. There's about 40 stations where we use a midwater trawl to go out there and collect. We're actually going for small fish. but we quantify everything in those trawls. And this is an example of the catch for the first 2 years, or like 2011 and 2013. And what I want to point out is that little number on the upper corner, which you can see is 0 there. That's the average catch of pyrosomes. So you can see it's been 0 throughout the whole sampling area. And I think if you did this probably 50 years before that, you'd probably see the same thing. Pyrosomes have never been reported up in our area before. However, starting in 2014, we started getting them in the very southern part of our survey area, just basically off the north of the California border. Then in 2015, 16, they started building up again. You can see the mean is going up dramatically. But look what happened in 2017 and 2018. Just orders of magnitude more. I mean, literally at every station we sampled, we were getting close to 50 to 60,000 of these in like a 5-minute tow with this net, which is and the thing that was amazing is that it totally swamped out everything. We were, we were getting so many of those. We had difficulty counting everything else because they were relatively few compared to this. It was just an amazing amount of pyrosomes.
Rick, can I interrupt?
Yes.
And ask a question? So the 2014 sampling season was when?
Oh, these are all around end of May, beginning of June, the same time of year every year. Yeah.
So just preceding
Just-
that.
right.
Period.
And you can see that they were down in the southern part, which that buoy was probably right here, so it's off of Newport, so they're probably coming in from this angle. So that's why we just started picking them up a little before the blob arrived. And then, so expanding that, so what I just showed you were the stations at the top of this graph in blue here, but the Southwest Center, a group out of the Santa Cruz lab, another NOAA lab, have been sampling the southern part of this range all the way down to the southern, to the California-Mexico border down there. And they've been classifying over even a longer period of time what their trawl surveys. So you could see that they had a number of surveys back in the 80s and stuff like that. And occasionally we catch some here in Central California. They catch a few of them, but very rarely. And look what's happened not only in Central California, but also in all the areas. It just took off the last few years. Dramatic increase. And the blue and red at the top there, those are the Oregon ones, so they're the highest of all of them. And I'll show you a plot. Now this, instead of numbers, this is biomass, so the total weight of pyrosomes in these trawls. And again, this is a log scale over here on the side. And you can see for the first 2 years, 2013, 2014, they were pretty much down off of California, just starting to get into Oregon. But then they started moving up 15 and 16. The line indicates sort of the center of their abundance, north-south, and the star indicates where the highest catches were. So you can see they're really moving up into our area. Looking at 2017 and 2018, they were really the dominant thing up here. Starting to drop off even down off of California in 2018. And in 2019, an interesting thing happened is they just about completely disappeared up north. We got one in our survey, but they got a lot of them still down in Central California, so they're not that far away. And just to kind of show examples of some of the biomass. So again, this again is a log scale on the right, but you can see that the biomass in Oregon and then the Blanco region, which is Southern Oregon, Northern California, was by far the highest. California really didn't change too much for either the central or the southern part. But then you can see some sort of interannual variation, especially in our area where a big boom and then it's gone. And then this showing the size differences, you can see that they're actually bigger up here than they were down in California. And I think a lot of this is because they're getting very, especially in this area, they're getting very small individuals coming in, which brings down the mean size. Okay, so now even looking at a bigger picture of the whole North Pacific, we went through and looked at other surveys besides the one I just showed you that also are out there sampling. And the one on the lower right is our Hake survey. The one on the lower left is one that the Southwest Center does for coastal pelagic fish like herring, anchovies, sardines. And then there's one in the middle off of Canada and then one up in Alaska. And all of these are showing high numbers of pyrosomes throughout there. And in fact, the ones up in Alaska continue to go even beyond. This goes all the way up to the northern part of Gulf of Alaska, but they were found even further west than that. And if you did a sort of a straight line distance between there and their normal distribution, it was about a 1,500 mile range extension. So it was a really dramatic change in the distribution of these guys. Okay, so now I'll talk a little bit more about their biology. There haven't been very many studies. In fact, that made it easy to catch up in the literature because there's not a whole lot out there. It's probably like 20 or 30 papers written on pyrosomes, which is amazing to me. But we do know this. There have been a couple studies that have shown that they eat fairly small organisms, and I'll describe that here in detail. So what pyrosomes actually are colonial animals. They're not, so what you see, the body of the pyrosome is actually a bunch of individuals that are genetically identical that form a colony like that. And what the colony is made of are these are the actual individuals, these called zooids. That's what they look like under a microscope. And you can see here's sort of a cross section of the body. So it's like there's a cavity. So there's so the water comes in from one end, and they actually pull the plankton and things out of the water, and then the water goes out the other side into here, and then so they use the same mechanism to both feed and to propel themselves in the water. So it's kind of an interesting arrangement, but there can be anywhere from hundreds to thousands of these individuals called zooids within one of these colonies. And again, here's another example of how they feed, where the stuff comes in and then the water gets expelled out to the side. So it's a very interesting arrangement, but they can actually move pretty quickly because of the water pressure that they push out. So one of the things that we were wondering, we wanted to look at this more in detail, and this is not typically the kind of thing that National Marine Fisheries Service tends to fund because they're sort of out of our area of expertise. So what we wanted to do is kind of collaborate with others who do this kind of work normally. So we ended up by working with Dr. Kim Bernard on campus and a bunch of people from here. We put in a proposal to the National Science Foundation, and we got funded to actually study pyrosomes. So we ended up having 2 years worth of funding, and it was able to fund some students and some of the researchers here at Hatfield. So the things we wanted to look at, we're sort of looking at we looked at the large scale distribution. We wanted to look at fine scale vertical and horizontal distribution and what is regulating that in terms of temperature and things like that. And also we wanted to look at their diet composition, and we did that a number of different ways, which I'll explain in detail here. So first, kind of looking at, we had a number of cruises where we were we were out sampling other things, but we caught lots of pyrosomes. And this is an example from a cruise that was in, where the one on the left is showing the sampling pattern for a zooplankton cruise that was in May of 2017. And so you can see the station coverage there. And instead of big trawls, in this one, they were using spinner plankton nets, so like a bongo trawl or a vertical net, which makes it easy to quantify the number much more than a trawl. And also it's good to catch specimens in good condition. So here's some results from, so what we're seeing here is the size distribution between February, which is kind of the blue, and May, which is the green. So you can see that over that period of time, they actually doubled in length. So they're growing pretty rapidly out there over a period of 3 months. The interesting thing, though, is that the average abundances were very similar between the two, almost identical. So it means that there probably isn't a lot of new recruitment or loss of the population, or at least those two are balancing out if there is. So that was kind of interesting. So looking at these different stations, this is a plot that shows the densities during 2 cruises. The May are in red, and the August are up in blue up there. And you can see that there were significant relationships between the abundance, which is along this vertical column, and then sea surface temperature, salinity, and fluorescence. And actually the fluorescence was not significant, but the interesting thing was that there was a real strong threshold that at really high productivities, we caught none of these. So they seem to not be able to do very well in the normal high productivity water that we have here. But certainly temperature, there was more at higher temperatures, and salinities up to a certain point, and then it dropped off. So there does seem to be some factors that are related to these. But the temperature and salinities are probably not typical of what they find here. It's more like what they're in their normal tropical habitat. The other thing we did was we wanted to look at the vertical distribution. So we teamed up with people from the Oregon Department of Fish and Wildlife who have this sampling device, or it's actually a stereo camera system, where they lower it down. We're looking at fish populations on the reefs off of here. But we had a student who was a hauling student, Olivia Blonheim, who went out with them and wanted to use this system to look at the vertical distribution of pyrosomes, because we know almost nothing about that. There are reports of them being found as deep as 700 to 1,000 meters in the tropical areas, but we know nothing about it in the more temperate areas like ours. So they actually did 8 deployments on this off a Stonewall Bank just off the coast here, shown here in the plot. And this is kind of the average of the vertical distributions found there. And one of the things that shows up, there's very few up in the surface. These are all daytime though. Very few pyrosomes up at the surface. They're all pretty much concentrated in this layer between 25 and 35 meters, which happens to be the depth that we throw that midwater net at. So that's probably why we caught so many. But and then they kind of taper off down deeper water. And the interesting thing was there wasn't much structure to the temperature or anything like that, so that didn't seem to be affecting their vertical distribution. We also, because it was a stereo camera, we were able to look at sort of their orientation or their tilt angle in the water column. And so examples of those measurements on the bottom plot figure there. It turns out that they're all pretty much, they're going in all different angles. Very few of them are vertical with a column like that, but they're going in a lot of different directions. But about 30 degrees seems to be the most common. So they're kind of hanging in the water like this, possibly moving up and down a little bit as they go. And then we did another set of experiments also off the coast, and actually that should be in a depth of 160 meters. Sorry, it's wrong, but there's a figure of what they look like underwater there. And then it was very difficult because of the high numbers that we're seeing here to be able to quantify them, but almost all of these were found in this blue area between 49 and 68 meters. And then you could see relative to temperature, salinity, and fluorescence that there really didn't seem to be anything driving that distribution at that depth. So I really don't know. They certainly avoided the very productive area above that with a high fluorescence. So that's, but they could be found anywhere else. But we don't really know what's causing this distribution to be like this.
So
okay.
Yeah. Just a quick question. You mentioned that daylight. Did you do anything at night?
No, we did not. We did not get a chance to. Unfortunately, for the GoPro, we didn't have the right lighting system. We have seen them, though, at other times. They do come up to the surface. We know that. And certainly in the tropics, they do that. They come up very close to the surface. So we have a feeling that that's probably where they are. But we weren't able to do anything with the video stuff.
Is there anything in the literature about migration?
Yeah, they do seem to do, there's been some studies that have shown they migrate at least 100 meters in a day, which is pretty good for an animal that size to move that much. Thanks. So now we're going to be switching gears and talking about feeding of them. And as I mentioned, there haven't been a lot of studies, in part because they eat particles that are very small that you cannot use a regular microscope to see. So we've had to use a number of different techniques, including biochemical tests to see what their food is like. And one of the common things that people use are what are called fatty acids. These are things that are found in all living organisms, some of which can be synthesized by organisms, but there's a group of them that are called the essential fatty acids that they have to get from their food. So if you measure the fatty acids of an organism, you can tell by their composition what types of food they ate. So it's a very common sort of tracer that they use in ecology to look at what the diets of things that you can't tell just by looking at their stomach contents. So this is just an example. There are a lot of different fatty acids that you can use. And then this figure shows sort of an example of these are sort of a lot of the base production things such as algae or diatoms or vascular plants. And you can see that they have different signatures. So you can use these different combinations of fatty acids to determine what something's been eating. So we did that. We took, or not me, but one of the graduate students, Hillary Sorensen, did this as part of her master's thesis. She went and dissected these and then processed them and put them in a mass spectrometer. And then we ended up with some interesting information from that. So this is showing across the top here are the different groups. So this is by month in inshore, offshore patterns. And these are different groups of potential animals or plants that they feed on. And you can see that there are a couple things that they seem to concentrate on. The dinoflagellates, which tend to be very small plants in the plankton, and also bacteria, which is not surprising because other people report it, but there aren't a lot of things at this size that can eat bacteria. These are really fine particles. They don't eat diatoms, which is our normal main phytoplankton out here. They're probably too big and probably not suitable quality for them. So this kind of gives us an idea of that they're probably eating just a few prey, different types of prey. They also, there's a way of looking to see if they're actually carnivores, eating other animals, and that got very low, so they're probably not eating any, they're just eating things that are at the base of the food web, not any other animals. Another way to look at it is there are a couple of advanced methods to look at it. You can look at flow cytometry, which is a method they use that to look at sort of blood count, cell counts, but you can use it to look at sort of the size particles that are in the stomachs of these guys. And you can see in this example up here in the upper left-hand corner, these are 1 micron beads that were put into the sample as sort of a standard. And you can see that they're eating things smaller than 1 micron. So that's really, really tiny animals. And the things that show up are things like small bacteria and very small plants that are showing up there. And then you can also do what's called environmental scanning electron microscopy and seeing actual animals. You can see the phytoplankton there and the bacteria there. So these are very small things that you would not be able to detect with a regular microscope. Okay, so then another technique we use is what's called stable isotopes, where you can also take tissue from an animal and use that to determine what it's eaten in the past. And there's 2 types of stable isotopes that are commonly found out there or commonly used. One is for carbon and then nitrogen. And they have 2 different things that they tell us. The carbon tells us where the base production, where the base of the food web is for that animal. So, you know, several trophic levels down, for instance, in case of a tuna, where the food originally came from. And that changes very little between trophic levels. So that's good for looking at where the food came from. The other one is nitrogen, which tells you how many trophic levels it up. So the animal is feeding at. So you can see that it increases about 3.4 parts per mil every trophic level. So we can use those 2 things to tell us a little bit about the diet of the pyrosomes. So here's a couple of examples looking at it by latitude on the upper panel and depth range on the bottom. And you can see that there are some patterns by both of those. So you can see that there are significant differences between north and south and also between inshore and offshore. But one of the things that's kind of interesting about these is this is sort of where your typical plant, the green is where a typical phytoplankton level would be, and about 3 above that is where a copepod would be, 3.5 or so, because they feed on phytoplankton. One of the things that you can see here is that almost all the pyrosomes are below even copepods, so they're really feeding a lot lower than a lot of the crustaceans that are out there. And in fact, some of them are even feeding below plants. They're feeding the bacteria probably in dissolved organic material. So these guys are eating very low on the food web here. Okay, so one of the things we wanted to do is sort of now, since we know what they were eating, some chlorophyll, not all of it, but they wanted to try to come up with an estimate of their ingestion rates and what their impacts might be. So we collected during that same survey, we collected a number of pyrosomes. We looked at over a 24-hour period. We looked at how much fluorescence is in their gut, so a measure of how much chlorophyll. You can see that it got a little higher starting around late afternoon and evening. There got more food in the stomachs than there were early in the daytime. And then we can use, so we could also look at how fast the food leaves out of their guts, which we need to know in order to figure out how much food they're feeding. So we're actually able to maintain them in these little plastic bags in the upper left-hand corner there. We had them out on the deck of the ship and just left them and sacrificed them every so often to look at how much fluorescence is left in their gut. And then we can use that to come up with sort of a grazing rate per individual per day. And we see this by different latitudes. And then we can compare what they are feeding on to what's out there, how much phytoplankton things are out there. And we see that it comes out to be actually not as high as we expected. It was somewhere between maybe 2 and 10% of the available food out there, which is a lot lower than other studies have found in the tropics. They found they're about 50%. And there could be a couple of reasons for that. One is that we're looking at only phytoplankton. If they're feeding on things like bacteria and stuff, they probably wouldn't show up here in the guts. And also, we're a lot more productive than these other areas, even during the warm blob. So there's a lot more food out there. So they're probably not having as big of impact. But 2 to 10% is still a pretty good impact for one organism out there on the coast. And this is just showing that same thing spatially for the 3 cruises, February, March, May, and September. And you can see the distribution on the top panel, the grazing rate, and then grazing impact. There are some areas, such as basically right off of Newport here, where there seems to be a really big impact. The other thing you can notice is by September, the population level is very low, so the grazing impact drops off dramatically. So what happens to all this biomass that's out there? We're seeing from the surveys, we saw about 200,000 kilograms per kilometer cube, which is really a huge biomass off the coast here. And this comes up to about, also about 5 individuals per colonies per meter cube. So an area about this size is going to have 5 animals in there. So that's pretty high abundance. So you got to imagine that that's a lot of biomass out there that's got to go somewhere. And also, there are people up in Alaska that measured the caloric content of these guys, and they're actually not that bad. I mean, they're about 5 kilojoules per gram, which is higher than some other invertebrates, but kind of on the same range as a lot of forage fish. So they're not that, they're not, I wouldn't say unnutritious. They're certainly probably average in terms of their nutrition, certainly higher than a lot of other jellyfish. So there's only really 2 places that this could go. It could either be, this biomass could either be consumed by predators, or it can sink down to the bottom and enter sort of the detrital pool and be, remineralized and decomposed. So there is some data, we didn't actually look at this directly, but there's a number of studies that have been showing that there are things that consume these pyrosomes. Example of sablefish stomach there to the left. A rockfish stomach from some of the ODF and W samplers. The stomach is packed with pyrosomes. And we even one report of a whale that washed up off the coast of Washington that had a ton of pyrosomes, both in its gut and in its colon. So it had a lot of, it was obviously consuming a lot of pyrosomes. Don't know whether that contributed to its mortality, but it's certainly not probably the best food for them out there compared to what they're normally eating. And this is some examples from the people in the FRAM group here I've been working with. Doug Draper here has contributed this figure. This is showing sort of the groundfish predation on pyrosomes based on their surveys of all 22 different fish species and around 6,000 stomachs analyzed. And you can see this is like this panel showing 2005 to 2014. These plus signs indicate that there's virtually no pyrosomes in the gut. There was one location where there was one, but that was pretty unusual. And if you started seeing 2015, you start seeing there are more circles there. 2016, even more. 2017, huge numbers. So there's a lot of predators that were out there feeding on pyrosomes, and it turned out that probably most of the ones that they were looking at were feeding on at least some pyrosomes. And there are a lot of invertebrates that were also feeding on pyrosomes. In particular, there's some in the water column, but also mostly on the bottom. And this is an example of a jellyfish. These are from some ROV videos. I was on a cruise up in, we went up and did ROV dives up in Quinault Canyon off the coast of Washington in August of 2017. We saw a lot of examples of pyrosomes in the water column, but tons of them on the bottom. And there's an example of a jellyfish that caught a pyrosome, another jellyfish that caught a pyrosome in the middle of the water there. But then when they got down to the bottom, sea urchins, sea anemones, crabs, other echinoderms, there were all sorts of things feeding on them. So they do get into the food web in other ways besides just consumption up in the water column. And then there's a lot of them that just probably sink to the bottom and just decompose down there. Examples from some ROV footage from ODF and W off the coast here. Another example from up in up Quinault Canyon there, and they tend to settle in sort of depressions. They move around, float over the bottom, and eventually end up there, where they'll eventually, most of them will decompose if they don't get eaten by predators. But the problem with that is that takes up a lot of the oxygen during decomposition, and that causes the water to get hypoxic, which has been, and in fact, this summer was a really bad hypoxia off the coast of Washington and Oregon. And we don't know whether it's related just to this, because we do get them without, we do get hypoxia without pyrosomes, but it was a particularly bad year for them. So it could possibly be related to all these pyrosomes dropping to the bottom and dying. The other thing that happens to them, and a lot of you probably have seen this, they wash up on the beach, and then they can be decomposed there or eaten by predators on the beach or whatever. So they do end up somewhere for the most part. An interesting thing, though, is that we never expected is that we actually saw some examples in our cruises where there were fish that were actually living inside these pyrosomes, which had never been reported before. The bottom one, medusae fish, those actually live on a lot of different kinds of medusae, so that probably wouldn't be unexpected. But the top one really was kind of amazing that we saw rockfish that were inside these medusae, which rockfish don't usually live with any jellyfish, so it was kind of interesting to see that. So this could be a positive benefit. I don't know why they would want to live in there, but it probably didn't hurt the fish. At least you can say that anyway. But there were some other negative interactions, and particularly with fisheries. There was a lot of examples, reports from fishermen and things. They would send us pictures of, where they, their gear, like for instance, the salmon fisheries, all their trolling gear was just covered with pyrosomes, and it really impacted the fisheries a lot in there. The one in the upper right is from a shrimp trawl. You see a little pink shrimp there, but all those brown things are all pyrosomes that were caught at the same time as the, and that really impacted the fishery because it meant a lot more time to sort these things out, throw them overboard just to pick out the shrimp. So, and there were a lot of fisheries that were impacted. You probably don't have time to get into them all, but nobody really has an idea of what the economic impact, but it probably was substantial. It also impacted our research a lot. This is an example down here of some people, Will back there in the background, who, when we were doing our sampling, we were trying to go out and sample, for, as I mentioned, for these little tiny fish. And we had to sort through these, totes of pyrosomes. It took hours and hours. And there were actually stations that we had to drop because either the net split or we just decided not to do them because there were just too many pyrosomes in the area. So it really did impact the research as well in a lot of different areas.
Rick,
Yep.
going back to the shrimper, if they were using lights to help lower bycatch, did that have any impact on the pyrosome bycatch?
I wouldn't think so because I'm not sure pyrosomes react to lights. I don't know what kind of sensory system they have, but I mean they obviously have something because they vertically migrate on a dial basis, but I'm not sure that they could swim fast enough to get away anyway if the lights were on the trawl. They're not like fish; they can just like move out quickly. So I would suspect not. But it's a good question. Okay, so kind of just to give an overall summary, this plot shows kind of like some of these what are called the mucus-feeding grazers that are out there. A lot of the things like crustaceans that we're familiar with, they sort of actively feed by using arms and things like that to bring food in. But a lot of the gelatinous zooplankton, particularly in this group, use mucus webs so they kind of put these things out or bring water through webs to filter feed, basically. And they're really important in the food web, but we don't have a very good appreciation for how much they affect food web. But the thing, as I mentioned, they can eat very fine particles, such as bacteria, that a lot of the other things, a lot of the crustaceans can't. And they also, can do, in some cases, have very big impacts on the phytoplankton populations. So they can end up by going to some higher trophic level predators, or in many cases, they have what are called jelly falls, that they fall down to the bottom and maybe get into the benthic food web, but probably most likely decompose. So they have a very important role to play in the ecosystem, and I don't think we really know enough about them now, but I think we're learning a little bit more at least. So just to kind of conclude here, as we saw, pyrosomes were found in very warm, less productive waters in the Northern California current, which may be more typical of their normal range down in the tropics. This area, as I said, this type of water is not usually found off our coast, but it was in the last few years, which may have led to this big increase in pyrosomes. They feed on very small planktonic organisms, have somewhat efficient feeding rates and high clearance rates, but we didn't see any, in terms of, and compared to what's out there, it didn't seem to impact as high as we would expect, but we still think that they could be very important, taking out a lot of the food that's in the system that could be going to other crustacean predators, say, that would, so this is sort of your typical food web. You have phytoplankton through small crustaceans and then up to forage fish, fish larvae, jellyfish, and then eventually up to the higher trophic levels. But I think what we're seeing in the last few years is sort of this alternate food web where it's going through very fine particles, going through pyrosomes directly up to some predators, maybe not all of them. So we're really seeing a completely different food web out there during this time. How does this impact the other phytoplankton or the other zooplankton? Really don't know because we weren't out there measuring their consumption rates. But one of the things that's kind of interesting, we do have some data. This is from that Trawls survey I showed you, one of the first slides for the pyrosomes. This is the catch of krill in those trawls. And so these are for like 3 years, 2011, 2013, and 2014, very high abundances. These are these mean catches, that's 175,000, so it's 3 decimal places above, really high numbers per trawl, way higher than most things. But look what happened once the blob came in and then the pyrosomes came in. Their numbers dropped off, several orders of magnitude, and pretty much were not a key player compared to the pyrosomes in the system. And these krill are very important for a lot of fish, seabirds, and mammals. So when you don't have krill out there, there's really a big shift in the ecosystem. So this could be, we're not sure if it's due directly to having pyrosomes out there or just a change in the water was not as good for the krill, but it's certainly been big changes in the ecosystem out there. So we do see that they could be important food for some consumers out there, but we don't know if it's the ideal food, and there are some things that didn't eat them. We don't know if they couldn't because of the size or whatever. Certainly some things like these forage fish are probably, they're feeding on fairly small particles, so they probably can't feed on something as big as a pyrosome. So that is totally, all the food that's going to them does not help the forage fish at all. So we think that there could be some real changes in the food web based on that. I guess the really big question is, will they return again in the future? As I showed you during 2019, there were hardly any of them off our coast, although I've heard reports that they were starting to show up a little bit perhaps in southern Oregon in the fall, but they were off of California, so it wouldn't be very difficult for them to come back. And there were some indications like earlier in the summer and fall that we would probably there looked like some indications that the warm blob might be returning in the North Pacific, although it seems like that's kind of died down a little bit. But a lot of the models that they've developed for temperature scenarios in the future are showing that we're going to be getting a lot more marine heat waves. In fact, they may be a permanent feature on our coast in 20 or 30 years. So we may be seeing pyrosomes. They may be not only a visitor to waters, they may be a permanent resident here in the coming years. So we may have to deal with a whole different ecosystem. So with that, I just wanted to acknowledge, in addition to all those people I listed earlier on, some of the ships that we worked with, and then also some of the funding agencies that helped fund this research. And then with that, I'll perhaps take any questions, and thank you. Okay, yeah, Gail.
How do they biomass?
They have bacteria that are commensal with them, yes.
The question I had was, dinoflagellates like warm water. That's why in the tropics, I wonder if there's been any population studies to compare since they prefer dinoflagellates.
It could be. Have you seen higher numbers of dinoflagellates the last few years?
I'm not counting them.
Oh, okay.
With warmer water.
Yeah, I think Zunin has been seeing some in their samples. I don't know exactly, but I think there's certainly been less diatoms during this because they require high nutrient. Yeah, it could be. I mean, there was a lot of food for them, and certainly up in our area because of the growth, you're probably getting a huge amount of dinoflagellates from them to feed on. Yeah, John.
And the pyrosomes showed up. Is that just the pyrosomes, or was it a pyrosome community that with all the other things with it? Was it like a whole biome that shifted? And then the other thing is that the fish that are eating the pyrosomes, are they things that are, or whatever the things are, do they normally eat them?
Okay, so the second question, do they normally eat jellies or pyrosomes?
Pyrosomes.
Well, a lot of these fish probably have never seen a pyrosome ever, so it's a very novel prey for them.
These are novel prey for these fish that you're looking at.
Yeah. There are fish in the tropics that eat pyrosomes, so it's not unheard of, but these guys, I'm sure. Now, some of those fish probably do eat salps. In fact, we saw even the forage fish, like in 2016, we did a study where we saw a lot of salps, which are much smaller than the pyrosomes, in the guts of some of the forage fish, which normally they feed on copepods and euphausids. So there was a real big difference there. So but the problem was that they were, we saw a big decrease in the mean body size of them. So don't seem to be growing as fast.
But
The forage fish.
was it a pyrosome ecosystem or just pyrosomes?
So are you saying that did other things come up with them?
Right, was it like a whole water mass that got north
Oh.
and things like that?
Well,
or
they
a
probably
different-
did. I don't think they swam on their own. They probably came up in this warm water mass.
Did they
And
come
you're-
with everybody else?
Well, we got other things like pelagic red crabs who came up during the warm blob, which we've never seen. I think the furthest north they were previously was like Bodega Bay or something in California. And they were off the coast and we had a lot of warm water species that showed up in terms of fish, which could swim up, I guess. In terms of plankton, I'm trying to think if there's any other things. Well, Bill Peterson saw lots of copepods he's never seen before. Like I think he had 16 new species that showed up during this warm blob that had never been recorded here before. So there was a lot of things that came up with the plankton. And then other things, of course, swam up in the warmer water. What's that? Oh, right, that's right, the octopus, yep. Mhm.
Jelly that ate flora.
Yeah, although those are normally found farther offshore here. They're not. Now that's the difference, that some things come up from the north, and then other things are found offshore like that in the by the wind sailors of Valellas. Those things come onshore when the water is warm enough or that the wind's going the right direction. So I think aquaria is probably, we've seen them even, probably the last 20 years we've seen some aquaria, but there were a lot more during this time period for sure. Yeah, Bill.
You mentioned very briefly about euphausids. In 1998, Sue Stolgor was doing sampling on the Newport line,
Mhm.
and her nets, when I was with her, nets were just totally clogged with salps.
Mhm.
And now, as I understand, those salps feed on slightly larger materials that are closer to the prey for euphausids.
That's correct, yep.
So, questions as far as where they're coming up and actually getting back to here. Are there more salps as well?
There were some of the salps, some of them actually declined, but we did get some like the Thades, that one that kind of, I don't have a picture of it here, but it has kind of like, there was one, I could probably find one. They have those funny little, they're quite
I see
big.
what you're talking about.
Those are big, yeah, those were higher numbers, but some of the smaller salps actually decreased in numbers, but
But as far as the euphausids population went, it was still robust then? Was it negatively impacted or not?
by the salps in 98?
I mean right now.
Oh, it's definitely been impacted, or it seems to be. It's not, it hasn't recovered compared to what it was, the previous years. So the interesting thing was they never really came back in 2019 when at least in our waters where there weren't very many pyrosomes. So I'm not sure what caused that. I mean, the temperatures were a little bit warmer than normal, but not the same as what we got during the warm blob and the El Nino, the marine heat wave. So I'm not sure why the euphausids haven't returned. Maybe it's a recruitment thing, that there's not as much adult population producing them, or what. I don't know. Maybe there'll be other people that might know that, but yeah.
Is there have you seen a size limit on the pyrosomes? I've seen pictures and they seem way bigger than I've seen on the beaches, and I don't know if that's exaggerated.
Okay, like this guy, for instance. I love this picture. This is actually that's a leatherback from Australia, I think, or somewhere down in the tropics. And there is one. It's not the same species, but there is one that grows to about 30 feet long. It's pretty amazing. And there's actually, you can go on YouTube and see there's like a diver swimming around, and he's actually swimming into the opening of the pyrosome. So, but fortunately, they don't eat things that big. But I don't really, that's a good question. I don't know, and I'm not sure we know. A lot of these things could be. I don't know. They add on. They actually add on like more zooids and just keep building up. So the original starts with a few of them, and it just keeps building up and building up. So it takes a while. Yeah, I think
they're
they're
genetically all the
yeah,
same.
they're just budding. Yeah, this one, and then I think they bud off new colonies or something. And I'm not an expert on their reproduction, so I'm not sure anybody's really studied it. But it's pretty amazing. But the 30-foot ones, I don't know. They might live to be more than a year or so, I would imagine. But I love this picture. It kind of looks like those, those noodles that the swimmers, to use to keep up. The turtle's just kind of floating around on his pyrosome here. Yes.
Kind of in that same vein, so the abundance numbers that you were seeing throughout the year, do you think they were moving away, moving south, moving offshore with that warm water, or were they dying?
Later in the year, they were probably dying, I think. And possibly because we did get a little upwelling in productivity, sort of at least in 2017, I think, in August, and that probably might have been too much for them to actually survive, and they probably get their filters clogged. That happens with a lot of these filter feeders. That's why they live out in really blue oceanic waters, because when they get close to shore, it's actually too much production, and they can't process it because the things like diatoms just clog up their gills and things, or their feeding mechanisms, so they can't survive. It could also have gotten pretty cold for them, but it could be a natural thing. They might die every year at that time. We really don't know much about them. And there's very few studies, as I said, I mean, nothing about sort of their demographics sort of in their normal range, so we don't know what they go through there. Definitely a lot more we could learn about these guys. Okay, John.
Is it normal to see things that bloom in the ocean and then drop dead and fall on the bottom? Do other things do that?
Well, regular
Fossils
jellyfish do.
that, and I don't know, other fish and things like
Well,
that.
euphausiids must, they only live a couple years, or either 1 or 2 years, so they probably have to die sometime.
But do you see dead bodies on the bottom coming?
Probably not, because things like euphausiids might be consumed before they sink very slowly. They might be consumed before they get to the bottom.
Whales, John.
Counts as squid.
Yeah.
Yeah.
Well, just regular jellyfish like, geez, sorry.
Not like that.
The regular jellyfish like moon jellies and sea nettles, they usually drop down to the bottom sometime in the fall or winter, and they can and there are places all over the world where they document these jelly falls, so they just completely cover the bottom, sometimes killing a lot of the benthic animals because they totally smother them. But it's just an incredible input of carbon to the bottom, food for all these guys. So it probably has its purpose. But whether that's the right kind of food for them, I don't know. But you got questions? Oh, okay, Mo.
Great presentation, Rick. Thanks.
Thanks.
Thinking a bit about the socioeconomic effect of that in our future, like those years must have been really bad for our newcomer fishermen and for the coast. And they're probably like really happy right now because, as you showed, the pyrosomes
Right.
are off a lot, so I think the shrimp are maybe back to form again. Do you think right now that's more just relief in that camp, or do you think did this like heighten their or like the awareness of climate change? Taking into account that, as you said, this is like just going to get way worse. Thinking about the next 5, 10.
Yeah, I think it is, and a lot of the early reports actually came from fishermen. They were sending me pictures of or somebody here pictures of what are these things that are showing up here. We've never seen it before. It would be interesting to actually measure the economic impact. There was a study a few years back where people looked at sort of jellyfish, Medusae in general, and it was a pretty big impact on even off of Oregon, the fisheries here. But something like this, I can imagine, it was huge. But I was trying to get fishermen, I was contacting some of the local extension officers or extension agents to get fishermen to send me information on what the impact would be. And I got a few people that said that, they lost a lot and they had to stop fishing in certain areas. But it would be really difficult to quantify that. But I think, it probably makes them realize that things are changing out there. They probably have seen that gradually, but something like this happening has been a really big change.
Yeah, if we would have that for more years in the future.
Yeah, we might have to adapt somehow, have some kind of a pyrosome exclusion device on our nets or something to keep them from getting caught.
Or come up with a fishery
for them.
Fish
Yeah,
them,
no, that's a good point.
Culinary,
creative
brainstorming.
I've actually tried them. I don't think they taste very good, so I don't know if we could do something with them. But
Jellyfish salad doesn't taste very good either, but it tastes like what you put on it.
There are places in the world that they eat lots of jellyfish.
Rings, instead of calamari rings.
I actually had a guy write me and ask me if they could be used for fertilizer because he was catching. I was up in somewhere up in Washington, and he said they were catching so many they were going to bring them back to use them for fertilizer. I said, "Oh, okay, no comment. I don't know what you could do with them, but they're probably too salty for that. But there must be something you could do with them."
They last a long time on the shelf if they sit on our beaches
Yeah, yeah. Well, there you go, something to do and start a company here. Well, okay, Miriam, one more question.
That was a great talk, Rick. I actually don't have a question. I just have a pyrosome fun fact. So prior
Oh,
to this, the
go
main
for
thing
it.
I knew about pyrosomes is they potentially brought America into the Vietnam War because a pyrosome was misclassified as a torpedo.
Really?
Yeah.
You
The
mean,
Gulf Tonkin incident started because of one of the big pyrosomes.
is that right? Because of the, was it because they look like,
My Zooplankton in college.
yeah, they probably, was this like acoustically they determined? Was this from acoustics? They could see pyrosomes in the
I
water?
think it was just, they started to get misclassified. People were finding pyrosomes that were bioluminescent, thought it was torpedoes in later
Oh,
war
really?
times, so the idea is perhaps that one was as well.
Big
Great.
pink torpedoes.
Pink torpedoes, yeah, a little suspicious. Was there any questions on
Slow
the?
moving one.
Just want to see if there's any, okay, no questions, all right. Well, thank you all for coming and appreciate it. Yeah, rockfish are eating a lot of them. Yeah, yeah. Oh, great. Well, we'll see you in a little bit. Okay. Yeah, they were. No, I bet you they were probably pretty good for rockfish because they don't move around that much. They don't need a huge amount of energy or anything. So, anyway, thank you. All understandable. Too fast. All right. So, okay. Cool. Yeah.
I think we all have to switch to study then.
Yeah.
In
Well.
10 years from now, there's not going to be anything else.
Oh, I know. Well, you got a lot of data from your Mexcal cruises. Is anybody looking at the pyrosomes from that?
Yeah, building just right now, I'm still finishing up the final library.
Mm-hmm.
But we made a lot of progress in our.
Did you see a lot? Did you see a lot of pyrosomes in it? Oh, yeah. Well, that would be the way to look at their
It's
distribution.
going to be the most we will ever know.
Ah, get going on it, man.
Right?
Yeah, you should do it. Well, vertical. Yeah, and plus you have all that temperature and other data that go with it. That would be that'd be the perfect way to do it. That's your next paper for you.
Well, you will know about it.
Okay, send me a copy. Oh, right. Oh, sure. Sorry.
Did you?
21. Oh, that's pretty impressive. Oh, yes. What's the capacity?
50.
Is it? Yes.
We're in Cambria and
All the ones on the edge, but we had people on the floor, and I brought them up to
Yeah, it seems like long.
Yeah, no.
But they're